Hi guys, I'm doing some demo videos since you guys can't be in the lab. First thing I'm going to show you is how to streak a urine plate, okay? Here are my plates. I have a blood agar, sheep blood agar. I have a McConkey agar. I have a urine that is quite old and it is on Tooth Fairy is the name of my patient. I'm going to label my plates. I'm going to put And I'm going to put the date. Really important to put the date. Today is what date? 30th of April, I think. 34. So I'm labeling both of them. If we were actually in a lab, you would put your accession number on here. The number is the most important part. I want you guys to see the plate. See this part here? This is usually where the inoculum starts, and we have them on both plates, okay? That's usually where we would start our plate. It's not imperative, but if you had an automatic plate streaker, that's what would happen. These are our calibrated loops. These will measure 10 microliters of urine as we put it onto the plate, okay? I'm gonna do this plate outside on the bench because I want you guys to be able to see the streak marks on it. Usually we would either wear eye safety or do it in our biosafety cabinet, which would be the most appropriate thing to do. Okay, so I'm going to gently mix my urine by giving it a swirl. I'm going to open it, and I'm wearing gloves because I'm handling a primary sample. I take only one loop, and I don't pull them all out and put them on the bench because these are sterile, okay? I'm going to dip my loop vertically into the urine. I'm not going to go too deep because if I go deep, the urine will cling to my loop and will come down and give me more than 10 microliters as I want. I'm going to go into my urine vertically, fill my loop. When I come out, I check that my loop is filled. Okay, it is filled. I do my blood agar first. I'm going to put it on and I'm going to drag my loop down, and I can see that my loop has emptied the urine. And can you see that there's a single streak mark down of the urine? Do you have a good angle?
No, that doesn't work.
Anyway, we'll carry on. Now I'm going to go very close together, filling up the whole plate. There we go. Can you see that? We're filling up the whole plate. I'm going to do it again with this one. Fill up my loop. I didn't change loops. I'm going to put it here. I'm going to go straight down. I've lost my loop, my drop. As you can see, the drop has gone onto the plate. Now I want to get it there, okay? There you go. So we have a urine that's, I've used all the space on the plate to fill it up, okay? Now I have to take my loop and immediately discard it. Then I quickly cover up my open urine because I don't want to smell piss all day long, all the time. So now I'm going to incubate these plates. And I also want to show you that handling plates. I always would handle the plates this way. I would never have plates sitting around like this, okay? I would never pick up a plate and say, hey, student, look at this, like this, because they might pick it up and, uh-oh, they've dropped the media. So we store plates like this with the media up and the lid down. And one of the main reasons for that is because we're going to get condensation from the media on the lid. We're going to incubate them in a nice warm place, and we'll get a buildup of moisture on here. And if this was like this, the moisture is going to drop back onto the agar and give us a swirly mess of bacteria and no individual colonies, okay? So that's what I would say about that. So now they're going to be incubated at 37 degrees and they're going to be incubated overnight. And then the technologists will look at them the next day and assess the colony count, and they can count the number of colonies that have grown on this plate because we've used a calibrated loop that will give us a colony count that will be in CFU per liter or CFU per mils, and it's colony forming units. Okay, a urine, again, so going over it, calibrated loop, mix and swirl. Plates need to be labeled before you inoculate them. You can use a sticker label on them. We have to make sure that the date of the media has not expired. Can you zoom in on our media here? Unfortunately, this media has expired on March 18th, 2020. This is Columbia agar with 5% sheep blood, and we have a product code. It's made by Oxoid, and there's a lot number and when it was made. Okay, that's the lot number. So this is the blood agar with sheep, and our McConkey has other things written on it. So as you can see, our media is all gonna have a label on it. These plates have all expired, but this is all I've got. Can you see the stuff all right? Okay. Okay, let's move on to another subject. What I'm gonna show you now is how to make a smear from bacteria. Okay, we're going to make a smear from bacteria, and I need some glass slides, and here we go. We're right here. We have a nice glass slide. We're going to take a pencil because we're going to label our slide. And I have a plate of bacteria over here that somebody has used. They touched their hand onto it, so I have a few colonies of bacteria. Can you see them?
Yep.
Yeah. There we go. Fingerprints, these are colonies. So it looks like Roanne washes her hands nicely, so that's good. So I'm going to make a slide. I'm going to use a white colony. So I'm going to put Roanne, the patient's name. So I would have a sample number. So I'm going to put Roanne, and I'm going to put number 121. Let's pretend we have a lab number. I'm going to put today's date, 34. I might put my initials on it, okay? So now what I'm going to do is I need a little drop of water or saline. I'm looking for, somebody's messed around a little bit in our lab and I can't find our transfer pipettes. Oh well, I'll figure it out here with my loop. Transfer pipettes are in this. Oh, here they are. This is what I'm looking for. Okay, I'm going to use my transfer pipette. If I had saline made, that would be better. I would use saline, but I don't have any saline made. So I only need a little tiny drop of water or saline on my slide. That's it. That's all I need. Tiny little bit. Then you take a sterile loop, or you can do this in the biosafety cabinet with the metal loop, and I'm going to touch my loop to the colony. Here it is. Can you see the colony? And I'm going to just touch it. Now, zoom in here, Liam. I'm going to mix it and emulsify it and spread it out a little bit. I'll show you what I've got here. Can you see?
No.
No, hang on. All right, I'm just there. This is what I've got. So now, what I need to do is I need to let this air dry, and then I will heat fix it. So I'm going to leave it on the bench, and I'm going to let it air dry. And then we're going to do a gram stain of that sample. Okay, now we're going to go into the biosafety cabinet, and I'm going to show you how I'm going to streak plates using a metal loop inside our biosafety cabinet. Okay? I need to just get in. Oh no, I'm not going to do that. Come over here. I'm going to take 3 plates. So this is our incinerator. As you can see, it's nice and red hot inside of it. This is my metal loop. I have a metal loop. And I'm going to put it in here and I'm going to let it get red hot. I'm not going to walk away from it because it gets so hot that when you touch it, you say, ouchie, wah-wah. And that's not good. So now, here we go. We're going to take it out. You can see red hot. Did you see that? It's only red hot for the second I take it out. It's slightly red when I put it in. Okay, so I'm going to heat it up, and if I was doing a swab, I would have a swab. I'm going to show you the manufacturer swab. This is just a temporary. I have a big swab. I touch somebody's, the doctor or nurse collected a sample. I have to take that out. If I leave that in, it will get so hot that it will burn me when I touch it. So I have to. Okay, so I'm going to take my swab. And remember I showed you this area on the thing? This is where I'm going to make my inoculum. I'm going to take my swab. I can see where it is with my finger. And I'm going to put my swab on it like that. And now I have to make an inoculum area using a circular motion, and I need to make sure that I roll my swab. Roll it, because if I touched a pustule on one side and I never rotated it to the other, I might not inoculate the organisms onto the plate. Okay, so I'm doing my first one. Now I pick up my second plate. I'm going to set up my second one. I have an aerobic and an anaerobic plate. And then I have them a combi. These plates would all be labeled with your patient information before you start this process. Swabbing it, swabbing it. And if this was a real patient swab, I would put it back into the container. And I'll show you those containers later, okay? So I'm getting rid of that. So now I'm ready to streak my plate. Okay? Just adjusted my wire a little bit. Now I'm going to grab my other one from over here. I'm going to have 2 on the go at the same time. Okay, just checking my wires. So what I'm going to do is I'm going to get one going, and then I'm going to streak my plate, and in between quadrants I'm going to switch, and I'm going to use the other clean loop. So here we go. I'm going to streak my plate and touch it to the agar to make sure it's not too hot. Can you see it? And I go close together for that first quadrant. And then this loop is dirty, so I have to sterilize it. Here's my clean loop. I'm going to do it again for my second quadrant. And you notice I've gone in a few times and I've come out. So now I have to switch loops again. Here's my, there we go. Touch it, make sure it's not too hot. We go in a few times. And now I only have a little, very little bit left on my plate to do. And here's my last quadrant. Just a little sprinkle. Now I'm going to do my next plate. I'm going to do it a little quicker. Okay? Ooh, my lips are getting hot. First quadrant. Ooh, hot stuff. Second quadrant. Oh, this is unbelievable. Third quadrant. Flaming loop. The important thing is to rotate the plate, because you could get a lot of serious carpal tunnel if you're twisting the wrist the whole time to get it right. Okay? Now, I want you to zoom in problem, and you're going to slice into it. But that's normal for beginners. So I want you guys to see how each plate doesn't look the same. They're a little bit different in their streaking patterns, and I'm going to pull them out and lay them. But it's almost like a signature that people have when they do this this process. I can look at people's plates and know who's streaking great. Okay, so I'm going to pull them out and we'll look at them and sort of critique my streaking out here on the bench now. Okay, so let's see. There. So there's the first quadrant. You see the inoculum. The first quadrant is close together. The second quadrant here on the side, I've gone in a few times to the heavier streak line and pulled it out and come apart, spread apart more. The third quadrant is this one here where the light is hitting it. Again, spread out even more, and that fourth quadrant is just a little squiggle. Okay, that's one. This plate here, this is the first quadrant. My inoculum is about the size of a loonie. And then I'm going here is the second quadrant. It's kind of hard to see this one. Second quadrant. Yeah, this plate's hard to look at. Let's look at the other ones. Here's my inoculum area, my first quadrant, second quadrant here. That's a good one. Third quadrant, I've gone in only 2 times, back into the second, and dragged it out. The fourth quadrant, you can see where I went into it, 1, 2, and maybe one little bit more, and just a little squiggle. And I've tried to use all of the media space to fill up my plate, okay? And that's a decently streaked plate. It should give us good isolated colonies. Now, my slide should be dry already from when I made the gram stain. Let's go get it and have a look at it. It's not quite dry, almost dry. So I'm not quite ready to fix it yet, but I'm going to do it in a moment. While that one is drying, I'm going to show you what a regular swab looks like. This is what the regular swabs look like. This is how they come. I'm going to open it up and show it to you. These are the swabs that we would get from our outside doctor's offices, and they would take them and send them to us. It could be a throat swab, whatever. These are sterile when we open them. This is the container. It has not got a swab in it, but it has media in it. This is media to maintain and preserve the integrity of the sample and not to allow it to grow or flourish, okay? This is the swab. I'm going to take a swab of my cheek and I'm going to make a gram stain of it. Okay, here we go. You could do a throat swab or an ear swab or something, but this is as adventurous as I'm going to go. So now, look, I'm using my pinky. I'm going to uncap it and I'm going to put it back in to my swab. And then the nursing or the doctor should label this sample. Okay? I'm getting rid of this, the cover. And now I'm going to also make another stain slide of this sample because this is a primary slide. I do not need saline to make this slide. I do not need saline. I'm going to take my swab out. I would be in the biosafety cabinet, but for the purposes of this, I'm not going to be. I'm going to take it in and I'm going to roll it on the glass, and then I'm going to take it and I'm going to go like this. I have to make sure that it's labeled. And I'm gonna say mouth. And I'm gonna hold it up so you can see. I just wanna look, no, I wanna look in and see how it's looking. I want you to see. It's hard to see it. There. There, so you can see. So this is a direct smear made from a sample. This is different than the smear that I made over here from the bacteria. This one from the patient, I did not need any saline. As it stands at the moment, it could be infectious because it has patient cells and material on it. This one is regular old bacteria in a suspension that is not likely to be infectious, okay? So this slide is pretty much dry and I'm gonna heat fix it. Let's go back to our heat source in the biosafety cabinet. My patient sample, the one that I just did of my mouth, needs to air dry. So I'm gonna sit it in here and allow it to air dry. Once it's air dried and we can look at it and see it's not wet anymore, I will heat fix the slide. This one, our patient's smear from the bacteria is now dry. It's got a little bit of moisture. I should wait for it to dry, but in the purpose of this video, I'm gonna carry on, okay? Can you see where the slide material is on this fix? Okay, so now I'm gonna heat fix it. So this is what I'm gonna do to heat fix it. I'm gonna hold it to a heat source for a few seconds. And now I'm gonna check that by touching it to my hand, it's hot to touch, and that's good. This is a fixed slide, okay? So, is my patient slide dry yet? Not quite. I'm gonna come back to that, okay? Now we're gonna get set up to do our gram staining, okay? Our gram staining, and I think you need to pause it for our gram staining.